Hey, finally got the video to work. I've been trying for a while. Anyways, this is Biology 24.2, and it is our last section of the biology book. And I'm kind of bummed that I didn't get to teach you guys chapters 22 through 24 because they are definitely tough chapters. And tomorrow I'll go back and we'll do Punnett squares just so you kind of understand that. A lot of you think I don't really know Punnett squares, but actually there's probably some questions on your ACTs and things like that about Punnett squared. So I want to make sure you get that. Um, 24.2, and it talks about the central dogma for molecular biology. And if you look up the definition for dogma, it basically is talking about something that's not necessarily all proven, but it's widely accepted authoritatively. And that's basically what molecular biology would be. This is what we believe things to be because it's such a small and like how they study and that kind of thing, and all that. So anyway, three different processes of in the cells that we're going to talk about today, and you have replication, transcription, and translation. All right, so replication is basically just the DNA making a copy of itself. And so if you like put the DNA on a copy machine, it makes a copy of itself. That's replication, right? Kind of easy to think of it that way. And then you've got transcription. This is where the cell copies information contained into the DNA and it puts it in to the messenger RNA. We're going to talk about different types of RNA. The RNA is basically just a kind of a copy of the DNA. And the messenger RNA molecules in turn provide the information to the ribosomes to make proteins, and then that process is called translation. So let's break it down a little bit farther. So before a cell divides, it makes a copy of its DNA. So it is before it's dividing is the key there for replication there. So it first has to unwind or unzip itself, and you can see what it does on the bottom of page 491. Look at that paper, or look at that, and you'll see how that works there. And so it kind of unzips itself before it makes that copy. As it zips itself back up, it's really important that it has the base pairing still the same, and all of that. The adenines and thymines, the guanines and cytosines, all of those are going to be checked for errors before it's zipped back up. Make sure that they're pairing together properly, and so on and so forth. So replication will result in two identical DNA molecules whenever everything's said and done. And that's important there. So then it goes through transcription. And if you look on 491, you'll see that very first paragraph is really important about transcription. This is one of the most important purposes of RNA is its role as a working copy of the DNA blueprint. So if you think about like whenever you build a house, you have lots of copies of the blueprint. You may have a master copy that you store somewhere in a safe and you don't want anybody touching it, or maybe they have it over at the hardware store or whatever. And then you've got, your contractor has a copy, and your plumber has a copy, and your electrician has a copy, and whoever, Uncle Jake has a copy. I mean, there's lots of copies. And that's kind of the idea there. You want a working copy of the DNA blueprint. And when a new protein is needed, the DNA is going to code for that protein to be copied into what we call messenger RNA, or a little mRNA. And the messenger RNA is going to be produced by transcription in the nucleus, and then it's going to be shipped to the cytoplasm, where then its ribosomes are going to be used to make a template for it to make proteins. So if I had a blueprint of something and I needed somebody to make it, I'm going to keep the master copy. I'm going to give it the copy of it to someone so they can go make whatever it is that I need them to make. And that's kind of the idea of transcription there. Page 492 at the top, look at the table 24.7 in particular with transcription there. After the segment is copied, the DNA is going to be zipped right back together, and it's going to be twisted back into its normal double helix shape. And so as it does that, it still has to pair with those special pairs, those base pairing there. So you're going to have uracil or thymine with the adenine, and then guanine always with the cytosine. Remember TA and then GC or CG, however you decide to remember that. In humans and eukaryotes, other eukaryotes there, it'll produce a pre-messenger RNA, and then that's helping it prepare for the translation instead of just messenger RNA. There. Then you go to translation. All right, so lots and lots of genetic experiments in order for them to figure out what this process right here it was. And basically it has a lot to do with amino acids and proteins. And so they discovered information is coded into the messenger RNA in what they call codons. And a codon is basically just a consecutive sequence of nucleotides. Nucleotides would be, like we've talked about in the nucleus before. And basically, those codons are going to be turned into with amino acids. They're given a special genetic code, and so it has all different kinds of codes, the different nucleotides. You can see that table 24.2. If this makes sense to you, you should probably be a genetic engineer. You should probably go into molecular biology if all of this is fascinating and it makes sense to you, because it probably is over the majority of people's, even smart people's head. But you can look and see the first nucleotide there has a letter that it's going to go with there. Well, you understand uracil, cytosine, adenine, guanine. That's what that first nucleotide would be. Then you've got the second nucleotide, and it's just got these codons, and it just depends on what protein it's making or what amino acid it may be making. And then again, you've got the third nucleotide, which is pairing with the first one there. So anyways, this is basically like if you think about coding in a computer and computer language and all that, this is like similar to that, but so much more complicated. And it's what is telling our cells what to do continually, this translation, basically. You can see the codon, up on the top of page 493 and what some of those would work out to be. Some of them are going to be transferred into proteins. Some of them are going to tell it when to stop. It's called a stop codon, and it just tells it stop making this protein. It has a start codon. It tells it to start making the protein. So not all of them are codons like that we're thinking of, like making actual proteins. The proteins are manufactured by the ribosomes. We talked about that earlier this month, and the ribosome consists of two subunits, which would be one large one, one small one, and they're going to be separate in the cytoplasm, but then they'll come together in the proteins when they're made in the proteins. And this would be considered ribosomal RNA. And so we've talked about messenger RNA, ribosomal RNA, and then we also have transfer RNA. And basically that's what it does, is it works with transferring things there. You can read a little bit more about that. The process of making proteins from that RNA pattern and all of that we're talking about, that's called translation. And like I said, one of the best ways, if you're looking at just the general understanding of the replication, transcription, translation, it's that very first paragraph on page 491. It tells you exactly what it does. Making a copy, replication, where the copy goes into messenger RNA, transcription, and then translation would be where it's making a protein from that messenger RNA, that blueprint, that copy of the blueprint there. Right, and we talked about the genetic code. Then you've got over on 494, it kind of talks a little bit about how it's making the protein synthesis in the cell, and the different types of RNA. Those are collectively known as small RNA. And then there's also non-coding DNA, and like I said, it is going to basically help determine chromatin or chromosomes, where they are, where they're going. They're going to splice off certain parts of cells. They're going to tell it when to start, when to stop, that kind of thing. So that is non-coding. It doesn't have that big code in it like we talked about a few minutes ago. All right, then bottom of page 494, mutations. And we've talked a little bit about mutations, and it's the random change in an organism's DNA. But it's really, really important because when DNA copies itself, it has to be extremely accurate. And obviously, it's an actual, if I were to give somebody a blueprint, but something smudged and messed up on the sides, then something's going to go wrong when they make whatever it is that they're making for me. If it's a house, stuff may not match up. You just never know. So it's important. It's important to for the cell's DNA to be copied perfectly, identically. So occasionally, there's little random changes in the DNA, and it's little mistakes basically, and these are called mutations, typographical errors, if you will. And they may result in improper base pairing, where it tries to meet up with another base that is not fixed, it's not going together properly. And anytime that you have a mutation, sometimes it's so small, so insignificant that you don't notice as much a change, but most of the time it results in a disorder or a disease or something along those lines. On the top of page 496, it says improper base pairs occur when a base is added, deleted, or substituted in the original DNA sequence, and the results are generally very serious. Physical damage to the DNA molecule can result from oxidation, radiation, or carcinogens, cancer-causing substances. And so then you realize, oh, okay, mutations are also cancers. They can be. They can be involved in one gene or deletion of many genes or chromosomes or something along that line. If the mutation occurs in the DNA of the organism's reproductive cells, it could be transferred to the parent or to the children, to the offspring there. When mutations damage the genes that control the cell cycle, for example, cancers, tumors may result as the cell divides uncontrollably. As the cell divides uncontrollably, it forms tumors, and that eventually will be found as cancer. Or sometimes not, but that's what that's talking about there. And so you can read on to talk about that. But DNA, of course, is our master plan for life. It's in every one of our cells. Deoxyribonucleic acid, and then the RNA, that's ribonucleic acid, it just takes off the deoxy part there. And of course, all of these are extremely complex processes, but it's really important for you to see and for you to realize how organized those processes are. And of course, the Lord even tells us in the Bible, let all things be done decently and in order. And even the cell cycles, even something so teeny, teeny, tiny small, has that same principle backing it, decently and in order there. All right, so you can do section review 24.2. These are going to be kind of difficult questions. So let's see if some of you all are actually watching these videos, right? And I'm going to help you a little bit with 24.2. And it says, why is DNA replication necessary in a cell? And why must it be very accurate? Well, we just talked about it has to be very accurate because the new copy of the DNA has to be an exact copy of the RNA so that mistakes won't be made whenever it's being transferred, basically. But why is it necessary in the cell? And that's because it has to make another copy of itself before it divides. It has to make another copy of itself before it divides. So each cell has to have a copy. So if one cell dies, oh, it's okay. I got a copy of the DNA. I know how to do it. That's kind of, the idea there. Explain the roles of DNA and three main types of RNA in protein synthesis. They're just basically asking that DNA is the master plan, the blueprints for the cell, and they use that to make the RNA. And then the three types of RNA, that's easy. You can find that on your own. How does the process of base pairing help ensure that the DNA is replicated and transcribed correctly, and only one type will fit into another? So as it's assembled, it's going to not fit, just like a puzzle piece. You can try and try and try, and sometimes it's just still like it just won't fit. And that's the idea. It kind of like is a siren for it to be like, oh, nope, that's wrong. It doesn't fit. And so that helps ensure that it will zip up properly. And then what is one way mentioned in the text that accumulated mutations might lead to the degenerative diseases that accompany aging? Make sure that whenever you see that, it talks about with aging in particular, not just disease, but it's clear on that one. And in what other ways might accumulated mutations contribute to aging? And there's, definitely things like cells are going to die. So it would cause deterioration in all of your systems, but especially in your nervous system because of the neurons, the brain, the spinal cord, that kind of thing. It's going to cause cells to malfunction, cells to wear down, and they don't repair themselves properly. All of that has to do with aging. So as we get older, not always that great. And then number 5, if you have it, why are mutations in somatic, that's body, cells, not transmitted from generation to generation? And that's because the genetic information is only transferred through the reproductive cells. And so only if it's going to be in the reproductive cells can it be transferred to the offspring there. All right. And that is it. For the most part. You're going to still go back and do Punnett squares. And some of you try to do that on your own, and that was good. I didn't really check to see if they were all right because I'm going to show you that. After that, the Punnett squares, then you'll basically just have your final exam to do. And as you do, you have an entire week basically to do your final exam. But your final exam is like 7 pages long. So some of it you can print it out if you want. But if you're near the school or whatever, I'm going to leave some of that. If you haven't already gotten that, I'm going to leave some copies of that test. You just pull that where it says biology, just pull that test out and you can take it with you. You can either bring it back to me either at my house or drop it off at the school, or you can send pictures to me. Just let me know what you're going to do and make sure it's still done on time by May 15th there. And you'll be done. Yeah. I miss you guys. I miss seeing everybody. I miss talking to you. I hope you guys have a wonderful summer, and I hope you can get out and about soon, and hopefully we can see each other soon. Talk to you later.